तू दीवाना ले रहे हो आज ₹1,00,000 के
मोटे करते छोटे करते हो ले गया मुश्किल है हाँ वो फांसे हटा रहे तो बाप आता
क्या
हमें करती ना उसे क्या होता है सीए खेत वाला फिर बाद आज नहीं होता
हाँ
मेरे बात तो बात करना मैं सीए तगीच माట్లాడుతాను मैं तो फर्स्ट दिन रहा मैं किसको आदमी बुलाया तो अच्छा लग रहा है संदर बुलाया इस बार लेके संदर को काम डर को काम से झाड़ा देखना मेरा नो मामा ने लगा मैं शेफर का कोला मैं लगा मेरा क्या मां अभी 5 बजते हैं तभी के खाने में क्या ला पाएंगे हम 8 बजे कुछ टमाटर की बीच का अच्छी है अंडा दाल को बनाया कहीं नहीं तो कर दो हाँ दाल खाई तो माँ नहीं कहती चुटकी तो बोलती है चटनी खाई तो मैं लेके आया
दाल नहीं सकती खाई नहीं
छठी हिती लो
किसी खाई दाल कहाँ है
और असली थी वो बा मैं देखे
क्लासेस कब स्टार्ट होंगे
कोई दाल ना रखी थी सामान मंगा तो मेरा होटल से मंगा अब जा तू आप पूछ लो तो सारा ये है बस चार और आज के खाने की बस
Students, please mute your mic.
Okay.
Three students, please mute your mic. Madam, is that you? Agri Challenge Academy? Hello? Anyhow, a very warm welcome to one and all students. I think this is today's demo class is on plant science, another important topic called molecules in molecular genetics. Another important topic. Our faculty will come and will take the class. Hope you are enjoying the classes. Thank you. Agri Challenge Academy, Agri Challenge Academy.
Why
ರೆಡಿ ಆ ಜೋಡಿ ದೇख ಇದು दे नहीं
एक रुपयाला हेअर ऑइल हा 2 रुपयाचं की घ्यायला
Students, please mute your mic.
म्यूट म्हणला
ये बस आता
Students, please mute your mic. All students, please mute your mic.
Good evening, students. This is Devdatta Panda. I'm a PhD scholar at TNA Coimbatore. Today, we'll be discussing the molecules in molecular genetics. Is the slide visible to you?
Yes, ma'am.
Please reply.
Yes,
Yes,
ma'am.
ma'am.
But your voice is not clear.
Okay.
Yeah, it's
Am I
okay. It's
audible
okay, ma'am.
clearly?
Your voice is
Yes,
yeah,
ma'am.
no,
Buffering.
somewhat better, a bit better.
Yes, ma'am.
Okay, thank you. And please keep your mic muted throughout the discussion, and keep your video shut so that it will not interrupt with the discussion. Okay. So before beginning today's discussion, I want to hear from you how you guys are doing, how you are feeling, because 2 days back only all the ranks and everything got released. So before starting the session, being your I am somewhat senior to you. So, being a senior, being a mentor, I want to say a few things. You never ever, whatever the result is, whatever result would be, never ever get disheartened. It is not late for anybody to start and begin. And there is a long career which is waiting for you in the future. So, don't just be disheartened and keep starting. Just identify what your mistakes are, what your mistake you have made, and just begin your journey from there itself and look for the future opportunities. Because if you keep resenting on the what happened in the past, you'll be trapped in the past, isn't it? So keep looking forward always, and be positive, and surround yourself with positive thoughts and positive energy, so that you will be whatever you are thinking, you can really achieve it, right? So with this short note, we'll be starting with today's session. Today's topic of discussion is molecules in molecular genetics. So you might be wondering what I'm going to discuss today. So out of the all the topics in the genetic section, there are basically various sections which are quite important, your population genetics, let's say your classical genetics and everything. But this time you might have seen in the questions, this time, not only this time, I have if you just see the questions from last 10, 20, whatever years you have, it is available somewhere from some memory based questions and everything. You will find out there is a significant portion of the molecular genetics sections they used to ask. So today my aim and my main perspective is not to explain the molecular genetics in detail, but we'll be touching the points from where you are asked the questions most in the exam. Since some of you might be appearing some of the exams in the nearby time, because not for some of the universities the entrances exams are over, and for some it is still remaining. So for them it will be very highly useful, and for others who are beginners, for you all, it will be a stepping stone where you can find a few new things for your revision purpose, like that. Okay, so let's begin with it. So what is the first thing that comes to your mind when you study when you see the topic genetics? Like in your day-to-day life, have you ever heard the topic genetics? Means for since you are reading studying the agriculture and everything, life sciences, you will be heard somewhere genetics. But your parents or your family members also, even if they don't know, or they don't study genetics, still you can find that there is a different use of the term genetics. Whenever you do something mischief or something, they used to scold you like ye to papa hai and all, ye mummy pe gayi. Nobody will, means they will say like he takes before you, like your mother can say, or he takes before him, your father can say, like that. So there comes the thing that we carry the genes from our parents. So there is a great link or connection between how we behave and how we are going to
Ma'am, your mic is not.
I'm extremely sorry. My internet was a bit fluctuating. That's why I got cut. So whenever we think of genetics, there we can see that the very first thing we study in the genetics is Mendel and the pea plants, isn't it? So the great Mendel, what the experiment he did, after that we got the section of genetics in our as a stream of the science, isn't it? So he is popularly called as the father of genetics. And another thing cannot be means we cannot study genetics without the help of Drosophila. So this is the queen of genetics. Why? Because most of the things we study and in genetics, and most of the hypotheses have been given based on the studies conducted on this famous fly, that is Drosophila melanogaster. And of course, the DNA, our favorite, everyone's favorite, DNA. How can we study genetics without DNA, isn't it? So all these things and moreover, like all of these things constitute a small little part of genetics. Okay. So this is the photograph I have shown you here. This depicts the royal family picture of Queen Victoria of England and his family with 4 kids, 1 son and 3 daughters. Why I am showing you this picture? This is has historical importance. Yes, it has some genetic historical importance. It has some importance in genetic history also.
Miss
Why?
Madam,
Because the Queen
Ma'am,
Victoria,
your mic is
Madam,
not
Madam,
clear.
she was hemophilic, and because she was hemophilic, starting from her, this disease spread to various through her progenies and her
Ma'am, voice is not clear, ma'am.
kids.
Madam, excuse me, students. Madam,
And their kids married to somebody else and
Madam,
somebody else, like that the
uh,
hemophilia disease spread to.
excuse me,
Any
madam,
problem?
please, your voice.
Ma'am,
Madam, your voice
your
is
voice is
not
not
clear. Please
clear.
check your phone. Madam. Yes, students, there is a severe network problem. The reason we are all in remote area, that's why we are unable to talk. Once this lockdown, everything was settled, everything will be fine. You won't face the such type of disturbance.
Hello, am I audible now, students?
Yeah, now it is okay, ma'am.
Yes, ma'am.
Okay, thank you so much. Again, I'm extremely sorry. My area internet connection is a bit poor. That's why we are facing so many difficulties. Okay, so what I was explaining is Queen Victoria was one of the initial figures in the history who had the hemophilia, and after her kids and wherever the ancestry went on, they had hemophilia. Okay, so hemophilia is famously called as the royal family disease also, isn't it? So this figure you can see here. This kid is the great grandson of Queen Victoria, and who is from Russian family. But since Queen Victoria's great granddaughter had married to this person, and she was the carrier of the gene that is responsible for hemophilia. So the hemophilia disease spread throughout the Europe. Okay, and after this happened, what happened is, they since the big person and important persons of the royal family become affected with this particular disease, so they emphasized on the research, what disease is this and how this disease is transmitting, and what can be the possible cure of that. So that gave birth to the initial research field in case of your genetics. Okay, and the initial researches were most of all focused on the human genetics sections. Later on, with the advent of various plant breeding scientists, and when they usually used to cross the things, and they began to think of the fact that how the various crosses are being formed, how the traits are getting transferred, all those things they wondered, and after that they started the research in the field of plant genetics. Okay, after that, this particular diagram shows you the very fact that how the disease moved from the Queen Victoria to the further throughout the Europe. Okay, okay. Then there are three important sections of genetics. If you just see, the first one is the transmission genetics, second one is the modification genetics, and third one is the population genetics. Transmission genetics greatly deals with how the characters are getting transmitted from one generation to the next generation. Here you can see the coat color of the particular cats, how from this particular mother, how these progenies are getting generated with various coat colors. And not only, for example, here coat color is mentioned, like that other traits also we can focus upon. Molecular genetics deals with the fact that how the hereditary material, our DNA, is being carried out, being organized properly, and replicated without a error. And if the error is permitted up to certain level also, how it maintains accuracy, how the DNA compactly organized, and how our gene expression things are carried on so that the organism actually survives. Okay, and the population genetics deals with the behavior of the particular population. It does not deal with the particular individual, it deals with the population. Okay, so these broad three things constitute the total genetics. So today we are going to see broadly about the molecular genetics approach. So if you just categorize, it is not possible in one class to deal with all the sections, so I have taken three sections. Basically, that is the DNA replication, transcription, and translation, and these three things popularly constitute a central dogma, isn't it? DNA replication is the phenomena where the one DNA makes the similar another copy of itself, and through a semi-conservative manner. And in the transcription, what happens is, from the DNA we produce the mRNA, that is the initial stage of gene expression, that the gene is getting transferred, the gene or the DNA stretch is getting transformed into mRNA. And in the translation process, the from the mRNA the protein is being synthesized. Okay, so all both these processes, transcription and translation, together constitute the gene expression phenomena. And all these three things together we call it as central dogma of molecular biology. Central dogma concept was given by Crick. As you that's the famous thing, and all of you know that Watson and Crick have contributed a lot to the genetic research. And you can see this particular thing, that is the central dogma of molecular biology, how the things are getting carried out, and that underlies how all these things behave, how they interact with each other, how the molecules produced from the replication, transcription, translation, how they interact with each other, how they carry out the body processes. That's what we are going to study about today. Okay, so when we proceeded with the study of this all these things, there in the field of molecular genetics, various ohms have been arose, and they form the broad aspects of the research. And what happens here is the first term is the genome. Genome consists of the total genetic material that is present in a cell, right? That is the genome. Second thing is transcriptome. Transcriptome means whatever the RNA produced at a particular cell at a particular time, that constitutes the transcriptome. Third thing is proteome. Proteome means all the protein that are produced in a cell at a particular moment of time, that constitutes the proteome of the particular cell. Fourth thing is metabolome. Metabolome is all the metabolite that are produced in a cell at a particular moment of time, that is called as the metabolome of the particular cell. Genome is the constant for all the cell of the body of a particular individual. It is constant, okay? But transcriptome, proteome, and metabolome, these things vary from cell to cell, and within a particular cell also, it will vary from time to time, okay? So we cannot say that these things are constant, but genome will be constant because genome is the DNA. DNA, there is no changes will occur unless and until there is a mutation. There is no change we can see in the genome. But the transcriptome, that is the which section will go for the from which section of DNA or whichever genes are going for RNA production, that thing we cannot say. Means to various responses to various environmental factors, different genes will respond, and different mRNA will form with different proportion, isn't it? Some genes will respond to drought stress, some genes will respond to cold stress, some genes will be responding to any other stress, and some genes will be there which are expressed throughout the lifetime, okay? They're called as your housekeeping genes, okay? So these are all about your various ohms in genetics. Okay, so when we start, what I told you about the molecular interactions, that's the basic molecules which interact with each other in the organism's body, that gives rise to various processes, okay, that give rise to that satisfy the various phenomena which need to occur in the body of the organism. First one is nucleotide protein interaction. Nucleotide protein interaction means you can see in the DNA what will happen is DNA has some motifs, DNA has some places where the protein can attach themselves, and protein has several protein has some active sites where they can bind to the nucleotide. And again what will happen is, so with the particular active site and with the finger like thing and the nucleotide with the binding part, they will interact with each other. And likewise protein and water, what will happen in the interaction of water and protein is, you can see water is the polar solvent, isn't it? So water will try to dissolve everything with it, but the things which are polar in nature, they can only solubilize by the water, and the things which are non-polar, they cannot be they cannot be dissolving in water. Apart from protein, nucleotide and water, there are other molecules like you can say various the metabolite that are produced in the cell. Metabolite in the sense, you can tell some of the toxic chemicals, some of the byproduct of various reactions, and they are used in some processes in the some processes and part of it, and some are toxic and body gradually eliminates through excretion processes, okay? So, well, how they interact with each other, I think I have already answered the question, right? So to begin with the first component of the today's discussion, that is DNA and the molecule associated with the DNA. So this is a picture which depicts the two sister molecules like DNA. This is the DNA and this is the RNA. Okay, DNA is generally double stranded and RNA is single stranded. This all of you know. And DNA is consisting of A, T, G, and C, and RNA is consisting of A, U, G, and C. Uracil is the difference between these two. And in DNA, you can find the complementary base pairing, which follows the rule that is given by Chargaff, Irving Chargaff. That Chargaff's rule is very, very important from an examination point of view. And they used to ask several kinds of questions They will just rotate the ratio and ask you the various kinds of questions. So whenever you're studying DNA and RNA thing, you need to focus upon that thing. That how means which ratio, the Chargaff's rule you need to focus upon. Okay. So the next thing is component of a deoxyribonucleotide. Okay. So in deoxyribonucleotide, you can see first thing is that it is a sugar molecule. This particular thing is a sugar molecule. This is the deoxyribose sugar. This question was asked this time in SRF, that in which particular carbon molecule of deoxyribose the oxygen was absent, and what is the which particular carbon is the difference between the deoxyribose and ribose molecule. So the answer is second carbon. In the second carbon here the oxygen is absent, but you can see in the ribose sugar, I will show you the next few slides. You can see here the oxygen is present in the particular molecule. Okay. And another question which is related to this, the students who are in MSc, they will understand this. Dideoxy chain termination, which is used for DNA sequencing. In that particular method, which is the extra carbon where the oxygen is absent. So that particular carbon is this one, 3' O. This oxygen is absent. So because of that particular region, what will happen is this 3' OH is the thing where the new nucleotide will come and join to this particular position. If this OH, this O is absent, means here no new molecule can attach. So the chain will terminate there and there. Okay. So don't worry, we will be seeing the particular topic separately. Just for your understanding or information sake, I told. So let's focus on the dideoxy molecule present over here. So this is the sugar molecule and this is the base. You can assume any of the bases like A, T, G, C, anything can be present over here. And this is the phosphate group. Okay. Here only, this is the first dideoxynucleotide. Here, that's why all the phosphates are intact and nothing has happened to them. But when the new phosphate molecule, when the new nucleotide is coming and joining over here, there will be two phosphate, two of these PO4 groups will be lost and one PO4 will be remaining here. And this particular section is the phosphodiester bond. So always and always remember that between what your phosphodiester bonds are formed, whenever your one nucleotide is getting joined with another nucleotide, that at that point phosphodiester bond is getting formed. Why it is called diester bond is because you can see here, this is the first, here one, normally ester bond is O, this side normally the ester bond is oxygen and both the sides some groups will be attached, that is the ester group, isn't it? Here you can see the oxygen is linked to the sugar over here and over here also, and here this particular group is, this particular bond is formed twice, this side also, that, and a phosphate molecule is involved in this. Because of all those things, this particular thing is called as phosphodiester linkage. Okay. Okay, let's move to the next slide.
No.
So here what I have shown you is how to study the particular DNA thing. So to study the DNA, we can go for so not only DNA, for any of the macromolecule, we can go for using the X-ray diffraction technique. Apart from that, we can also go for nuclear magnetic resonance and all the thing. That also we can do. But here I have, here this is the famous experiment which was performed by the scientist Rosalind Franklin, and what she has come up with is she used the X-ray source, and when she has passed the X-ray source through a narrow slit, then you can see the when the DNA was put over here. So when the X-ray film passed through the particular DNA molecule, it diffracted. The X-rays got diffracted, and the pattern produced over a film is captured. That film, that particular X-ray film which is captured, that gives the idea regarding the structure of the particular DNA molecule. Okay, so this is the diagram. You can see this diagram was captured by Rosalind Franklin, and from this particular diagram, what she concluded that DNA might be having some helical kind of thing. Previously, they used to think that DNA is not helical, DNA is circular or something like that. But after the particular experiment, they concluded and they were almost sure that DNA is a helical molecule. Okay, and this is the DNA polynucleotide strand. So what is happening over here is, you can see what is happening over here is, this is the sugar molecule, this is the phosphate group, this is from 5 prime here, this is the 5 prime and this is the 3 prime. And here why we call it as 5 prime and 3 prime, and we used to call this as 5 prime to 3 prime direction, because this particular chain, this particular sugar molecule, it attached to previous phosphate group through its 5 prime carbon, and to next phosphate group carrying the next nucleotide through 3 prime group. Okay, so always and always, whenever the chain growth will occur, the new nucleotide has to come and bind to the previous one's 3 prime end. Okay, that's why I used to call it as, so for this one 5 prime end is already occupied, so 5 prime end is not free up to this point. And if this particular, this after this there is nothing, let's imagine after this there is nothing, means this particular end will be free, isn't it? Like this, after this there is nothing, so this particular end is free. So they used to call this as, this particular in the very first nucleotide, the 5 prime end is free, and the very last nucleotide 3 prime OH is end is free. Okay, the prime and the letter indicates the carbon number. Okay, so that's why you used to call it as 5 prime to 3 prime direction. Why? Because the very initial nucleotide started, when it started, it has the 5 prime end free, and it continued the progression with the addition of the new nucleotide to the 3 prime end. That's why it is called as 5 prime to 3 prime direction. And DNA always and always grow in 5 prime to 3 prime direction only. Okay, and as you all know, there are two hydrogen bonds that are present between your adenine and thymine, and 3 hydrogen bonds are present between cytosine and guanine. Okay, so if somebody will ask you like, if you want to have the high melting temperature of the DNA, what you need to do? So definitely, since here 3 bonds are involved between C and G, so the DNA molecule which has higher amount of C and G groups will be difficult to break. So if somebody needs the DNA, if you want to engineer a particular stretch of DNA which has higher amount of C to G ratio, will have proportionately high amount of melting point. Okay, so here also the diagram depicts, this particular section is the phosphodiester linkage. Okay, next, coming to the various chemical bonds that are found in the DNA, this slide is very, very important, and from this particular section many questions have been asked in various examinations, like between sugar to bases, which particular bond is present within within the where hydrogen bonds are present, where hydrophobic bonds are present, what is their implication. All those things are frequently asked in the examinations. So between your what sugar, you can see there are covalent bonds present, that is the normal sugar, as in the present in the glucose or any other sugar molecule. So this covalent bonds present. Then the phosphodiester molecule, phosphodiester linkage will be present between the two subsequently joining nucleotides, and the hydrogen bonds are present between the two bases that are present, that are complemented to each other. The hydrogen bond will be present, and the hydrophobic interaction will be there, which you can see between the two strands. There will be several components which will be interacting with each other, and you can find the hydrophobic interactions also. Okay, so the coming to the features of the different types of DNA, here all these things you need to remember anyhow. This table is very important. Wherever we are finding the extended version of this table also, all the facts whichever is related to this particular table, A, B, Z, DNA, that you need to get through, means completely digest it. Okay. And means frequently they used to ask like how many bases are there per one turn, and what is the distance between two subsequent nucleotides, whichever has the flat major group, minor group, like that. And the whatever aspects that are related to this particular ZDNA, those are very important. Why? Because ZDNA is different from the rest of the two forms of DNA, because it is left-handed. So maximum of the features will be differing from rest of the molecules. Okay. So you need to see that. And the major groups, minor groups, they are important because they form the place where the other molecules can come and interact with the DNA and perform the any of the functions, like your whether the transcription will occur or not, whether other molecules will bind to it or not. And we are manipulating the things in this particular region for our genome editing purpose also. Okay. And one of the important feature which is not mentioned in this particular table is where the DNA is, these three forms of DNA are found. So B DNA is the normal DNA, you can find it in the normal condition. But A DNA is the DNA which is found when there is comparatively low humidity condition, around 75% of the humidity condition. Whenever it is there, that time you can find the A DNA. Z DNA will be found at very high salt concentration. At that particular condition, ZDNA will be found. ZDNA we can also observe when we are unwinding the particular DNA. As you can see over here, what is happening is, here you see the DNA is uncoiling here. The DNA is uncoiling over here. So wherever the DNA is uncoiling, in the nearby region, you can see there is this ZDNA is being formed. Okay. Just a second, student.
Madam,
What happened? What happened?
hello.
Okay.
Nothing,
Yes.
ma'am.
Any
Students are
problem?
requesting you should some Hindi in between them.
Okay. Okay. Sure.
थोड़ा थोड़ा just धीरे धीरे मैं हिंदी. They are asking some Hindi also explanation in between.
Okay. Sure. मैं करती हूँ. तो what they are saying over here is, जब भी हम
of next.
या जब भी हम ZDNA को जब भी हम नॉर्मल DNA को अनवाइंड करते हैं, उस टाइम हम क्या देख सकते हैं कि जहाँ पे वो अनवाइंड हो रहा होगा है, उस पर्टिकुलर रीजन पे कभी कभी ZDNA फॉर्म हो जाता है. ठीक है. सो नॉर्मल हेलिक्स यहाँ पे हम देख सकते हैं कि राइट हैंडेड हेलिक्स है, बट ये पर्टिकुलर रीजन में हम देख सकते हैं कि यहाँ पे देखो कि लेफ्ट हैंडेड हेलिक्स फॉर्म होके रखा है. सो इन दिस पर्टिकुलर रीजन यू कैन सी द लेफ्ट हैंडेड हेलिक्स ऑल्सो, but that is just to maintain the torsional stress. टॉर्शनल स्ट्रेस का क्या मतलब होता है कि अनवाइंडिंग के टाइम उसको सुपर कॉइलिंग आ जाती है, तो उसको स्ट्रेस लगती है. कोई भी रस्सी को हम जोरों से उल्टे तरीके से खींचते हैं तो उसके ऊपर से वो टूटने का चांसेस होता है. सो what I said is, whenever we are unwinding a particular rope kind of thing, there will be some sort of stress on the particular side. So to prevent that from breaking, what we used to do is it automatically induces the negative supercoiling. So उसकी वजह से हमें कभी कभार ZDNA देखने को मिल जाता है. ठीक है. नेक्स्ट थिंग इज हिस्टोन. हिस्टोन के बारे में सबको बहुत अच्छे से पता है. हिस्टोन इज वेरी इंपॉर्टेंट, एंड हिस्टोन इज द मॉलिक्यूल दैट इज वेरी अल्कलाइन एंड बेसिक नेचर ऑफ प्रोटीन. ये पॉजिटिवली चार्ज होता है, and this is positively charged. And what happens here is, here we can see 5 basic kinds of histone molecules, that is H1, H2A, H2B, H3, H4. Okay, 5 different types of molecules. H2A, H2B, H3 and H4, these 4 molecules, what they do, they constitute the core molecules. Okay, and the H1 molecule forms a cap over this. H1 molecule क्या करता है, ये चारों molecule जो यहाँ पे दिखा है वो सारे बीच के sections form करते हैं और H1 जो है उसके ऊपर वो cap के जैसा रहता है. Okay, so what happens here is it will form a cap and it will, this particular structure completely it will called as the nucleosome. Okay, and this nucleosome is a very basic uh, compaction module of the particular DNA, and further also we can see the particular thing. Okay, all the questions whatever you have I will answer everything, but I'll answer in the last. Why? Because this will break the flow, isn't it? So don't worry, put in the chat box, I will see everything and I will answer in the very last. So as I told you, there are two main classes of histones: core histones and linker histones. Core histone, this section is the core histone. Okay, and this particular section which links the one nucleosome to another nucleosome, that thing is called as the linker histone. You just see in this particular diagram, this is the one nucleosome. Okay, 1 2 3 4 4 proteins in double form. 4 proteins in double form they are present. 4 are visible in this side, 4 are in the opposite side. Okay, so here this is the histone octacore and this is the molecule which is present here like purple color, that is the linker molecule. Okay, so that's what I told you. So this point is very important, especially for the SRF people, like the core histones. In core histones, which amino acid is more prevalent, that is H2A, H2B, you can find more of the lysine, and H3 and H4 you can find more of the arginine. H2A और H2B के अंदर तुम्हें लाइसिन का प्रिविलेंस ज्यादा देखने को मिलता है और H3 और H4 के अंदर आर्जिनिन का प्रिविलेंस ज्यादा देखने को मिलता है. And the linker section, it has a peculiar characteristic that it will be around 54 molecules, 54 nucleotides, and the rest of the portion are 146. So together they form around 200 nucleotides per one nucleosome. Okay, and here this slide is very important. These two terms, that is the DNA histone methylation and demethylation. Okay, you can tell it as why these two terms are important. Why? Because histone is the molecule which is completely wrapped, which histone is the molecule around which the DNA is completely wrapped around. Okay, so whether all the enzymes can get access to your DNA or not, that depends on the tight compaction present over the histone. हिस्टोन है उसके ऊपर तुम्हारा डीएनए जो है रैप होके पड़ा रहता है. ठीक है, उसकी वजह से क्या होता है, से पॉलीमरेज या बाकी जो भी एंजाइम्स हो गए वो सारे आके तुम्हारे डीएनए के साथ इंटरेक्ट कर पाएंगे या नहीं पाएंगे, दैट विल डिपेंड ऑन द कंपैक्शन लेवल. ओके, कि वो कितना टाइटली बाउंड है, हाउ टाइटली इट इज बाउंड, दैट on that it will depend. Okay, so here you can see the particular thing, that methylation and acetylation. Methylation, the word itself is telling about the phenomena, that is here the methyl group is getting added. यहाँ पे मिथाइल ग्रुप का एडिशन हो रहा है तुम्हारे मॉलिक्यूल के ऊपर. ठीक है, और यहाँ पे एसिटाइलेशन मींस here the acetyl group is getting added to the molecule. Okay, so what is the here the besides the mechanism is not that much important, that okay where the histone where methylation is happening or not. Apart from that, the important point to note here is what is the impact methylation and acetylation. You need to remember this always, like methylation forms a mark, it will forms a flag kind of thing, and wherever high methylation is there, that particular region will be very tightly packed, and their transcription and all the things will not occur, cannot occur. Transcription and other portions, other enzymes factors cannot bind there, and genes will not be expressed. Okay, that's what will happen here. But the histone acetylation, what is happening over here is histone acetylation will loosen the packing, whatever packing is there because of the histones, it will loosen up that, and because of that the transcription factors can easily bind to the DNA and the gene will be expressed. Okay, so यहाँ पे क्या हो रहा है, डीएनए जो यहाँ पे पैकेज होके पड़ा था, आपने क्या किया यहाँ पे मिथाइल के ग्रुप ऐड कर दिए, ठीक है। मिथाइल ग्रुप क्या काम करेंगे, ये फ्लैग की तरह काम करेंगे। तो इसमें क्या होगा कि जहां जहां पे मिथाइल ग्रुप्स ऐड होके रखे हैं वो सारी चीज वो सारे जगह जो है ट्रांसक्रिप्शन फैक्टर के द्वारा आइडेंटिफाई नहीं होते हैं, ठीक है। उसकी वजह से क्या होगा कि वो जिस भी जगह पे मिथाइल ग्रुप ऐड होके रखा है दैट विल नॉट बी एनी हाउ ट्रांसक्राइब्ड और ट्रांसलेटेड, तो वो सारे रीजन हमारे क्या हो जाते हैं, दैट रीजन विल कंस्टिट्यूट द हेट्रोक्रोमैटिक रीजन। ओके, and you might have heard the two terms like euchromatin and heterochromatin. Euchromatin region, in the euchromatin region you can find less of the methylation event, and in the heterochromatin so euchromatin regions are always amenable to the process of gene expression, and in the heterochromatin region we can find very less amount of gene expression. Okay, एंड एसिटाइलेशन में क्या हो रहा है कि एसिटाइल ग्रुप इज गेटिंग एडेड टू योर डीएनए। सो यहाँ पे क्या होगा, जो पैकिंग ऊपर होके रखा था, अभी यहाँ पे देखो यू कैन सी ओवर हियर द पैकिंग, द डिस्टेंस बिटवीन द वन न्यूक्लियोसोम टू वन नेक्स्ट न्यूक्लियोसोम इज गेटिंग farther, isn't it? So in this place the molecule can come and bind, यहाँ पे कोई भी ट्रांसक्रिप्शन फैक्टर है कि बाइंड हो सकता है, जिसकी वजह से कि हमें फर्दर जो भी प्रोसेसेस है जीन एक्सप्रेशन के वो बहुत आसानी से हो सकते हैं। Okay, so because of this particular thing, because of the gene, whether the gene expression will occur or not, that will depend on the level of histone methylation and acetylation. And one thing I need to tell you over here is DNA histone methylation or acetylation, that is not constant all over the time. Okay, depending on the need of the hour these things change, keep on changing. Okay, so let's move to the next thing. Level of organization, this you might have studied from the very basic time whenever you started the studying the genetics, but this has important like the thing you can find the thing you need to remember is the dimensions I have mentioned over here. The first thing is the dimension of the normal DNA double helix, that is 2 nanometer. After that, when it becomes a solenoid kind of thing, nuclear means nucleosome structure, it attains a 11 nanometer structure. After that, when it forms a loop, it attains a width of 300 nanometer. After that, it becomes 30 nanometer. And finally, when we find the various scaffolds present over here, it is 250 nanometer wide. And at last, the supercoiled structure of the DNA in metaphase, you can find this is as 1400 nanometer. Okay, so you need to remember all the organizational sizes. ठीक है, तो यहाँ पे आपको सारे जो डायमेंशंस रखे हुए हैं कि डीएनए की कितनी साइज होनी चाहिए, वो सारे आपको याद रखने हैं कि किस साइज में कितनी होती है। वेरी बेसिकली द साइज ऑफ द डीएनए डबल हेलिक्स, the size of the histone nucleosome, and the final size of the chromosome. Okay, these are all about the molecules that are associated with DNA. Now let's see the molecules that are associated with DNA replication. Okay, so 3 types of basic replications, you know, including the prokaryotes and eukaryotes. In prokaryotes, you can see theta and theta model of replication and rolling circle model of the replication. I'm not going to discuss the mechanism over here. I'm just focusing on the molecules and how they're going to behave. Okay, because that sections are very, very important. So the molecular aspects only we'll be discussing today, not the processes. Okay, so among these 3 models, theta and rolling circle theta जो है वो आपको prokaryote में देखने को मिल जाता है, and the linear eukaryotic thing, this you will see in the linear, means since the DNA is linear in eukaryotes, you can see the normalization model over here. And the sentence what it tells is, here you can find the various differences between these 3 models. Here the DNA has to be circular because this is the prokaryotic thing. यहाँ पे circular होगी क्योंकि prokaryotes के अंदर तो nucleus जो है होता नहीं है, उसके अंदर nucleoid होता है. In prokaryotes there is no such thing called nucleus, isn't it? Everything is inside the cytoplasm itself. So the nucleoid whichever present in the cell, that is circular in structure, and it is linear in case of your eukaryote. And here breakage of nucleotide strand, here it does not occur, it just initially the structure is something like this, and from this what will occur is this particular strand will try to move away. Sorry, I may not find the time because because of time limitation I'm not explaining the model. Just remember, like in theta model there is no breakage of the nucleotide strand. In rolling circle, because one part has to replicate the other part, means here one cut will be formed from that particular region, complementary strand will be getting synthesized and the new DNA molecule will form. In linear eukaryotic DNA there is no breakage of the molecule. Okay. Okay, and essentially all these three molecules follow the semi-conservative pattern of DNA replication. There is no need to talk about that because all of you know there is whatever DNA prokaryote or eukaryote, they will follow the semi-conservative mode of replication. Okay, then what are the components and what are the components necessary for the bacterial cell replication? And will be similar kind of thing will be there. Some analogous molecules will be there in case of your यूकैरियोटिक सेल रिप्लिकेशन ऑल्सो. ओके, सो पहला चीज जो है यहाँ पे कि इनिशिएटर प्रोटीन व्हाट विल डू, ये go and bind to the origin and separates the strands of DNA to initiate the replication. It will go to the and bind to the origin of replication and it will start separating the strand. ये जाके bind करेगा आपका origin of replication के साथ और वहां से क्या होगा कि आपका DNA का strand separate होना चालू होगा. ठीक है, और किसके मदद से this molecule DNA helicase will help in the unwinding of the strand at the formation of the replication fork. This helicase molecule ये क्या करता है कि आपके DNA को unwind करने में help करता है. ठीक है, अब वो unwind हो गया तो उसके बाद वो unwind होके रहे और वो एक दूसरे के साथ annealing ना करे, इसके लिए single strand binding protein help करता है. This single strand binding protein what it does is it will make the single stranded DNA single stranded itself. It will prevent the annealing, re-annealing of the both the strands. ठीक है, then DNA gyrase, this molecules is move ahead of the replication fork, making and resealing the breaks. Okay, this DNA gyrase what it will do, it will move, it will be there before the replication fork also and after the replication fork also. It will make the replication fork and reseal the replication fork wherever it is formed. Okay, and the main function of this particular thing is to release the torque that builds up because of the unwinding of the replication fork. टॉर्क मीन्स यहाँ पे टॉर्क का क्या मतलब है कि आपका जो सुपर कॉइलिंग मतलब आपका अनवाइंडिंग की वजह से जो सुपर कॉइलिंग क्रिएट हुआ था उसके वजह से जो भी फोर्स क्रिएट हुआ उसको वो नलिफाई करता है. दैट फंक्शन इज डन बाय डीएनए गायरेज. सो व्हाट डीएनए गायरेज डज, द फोर्स व्हिच इज created because of the unwinding of the DNA, it just releases that particular force so that DNA will not break at that point. Okay, DNA primase, this will form the, this will bring the RNA primer and the short RNA primer upon which your new nucleotide will be synthesized. Okay, DNA polymerase 3 is the major enzyme. This is the main enzyme which elongates the nucleotide strand. DNA polymerase 1 is the enzyme which removes the RNA primers and replaces them with the DNA. So in case of Okazaki fragments, we'll see in a moment. So what DNA polymerase 1 will do is it will remove the RNA primer. Once the Okazaki fragments are formed, in the initial sections, the RNA primers will be there, so many numbers. So the RNA primers are removed by the DNA polymerase 1 and it will be replaced with the DNA at that particular place. Okay. Then looking over the DNA ligase, जो वो क्या करता है कि जो ओकाजी के फ्रैगमेंट्स वहां पे फॉर्म हुए हैं दैट विल सील द ओकाजी के फ्रैगमेंट. द कट wherever the cut is made, means wherever the sections of the Okazaki fragments are left hanging, it will just join the two Okazaki consequent Okazaki fragments. Okay, this function number one. And this particular table shows some other molecules like your topoisomerase. Topoisomerase is one similar to that of your DNA gyrase. Actually, DNA gyrase is a part of topoisomerase. Will be seeing in the next few slides. Topoisomerase also helps to release the stress of the DNA by unwinding and by causing the breaks and then resealing it. Okay, so topoisomerase क्या काम करता है कि जब भी हम लोग अनवाइंड कर रहे होते हैं तो ये जैसे आपकी एक मॉलिक्यूल हो गई, ये एक मॉलिक्यूल हो गई, इसके ऊपर दूसरी एक मॉलिक्यूल अटैच होके थी. ठीक है, अब आप उसको अनवाइंड कर रहे थे तो यहाँ पे उसको फोर्स पड़ा है. ठीक है, तो वो क्या करेगा, इस पॉइंट पे डीएनए को ब्रेक कर देगा ये स्ट्रैंड को अलग से ले आएगा और फिर से सीधा करके जोड़ देगा, ठीक है। तो उसकी वजह से क्या हो जाता है कि जो सुपर कॉइलिंग की वजह से टूटने का सिचुएशन आ रहा था वो नहीं होगा. What I told just explained just now is just imagine two ropes intertwined with each other. So what will happen is whenever topoisomerase is acting, what it will do, it will just to release the stress generated because of the uncoiling of the DNA. It will make a cut over here. It will release the particular strand. This strand will be released now, and again it will join the two cut portions later on, so that here whatever strain will be produced, that will be nullified completely. Okay, and already I talked about the other section, so I'm skipping it. So here you can see this is the main strand, this is the leading strand, this is the lagging strand. Okay, this is the parental DNA region, and this is the replication fork. Replication fork is moving in this direction, okay, in the 5' to 3' direction. And in this particular strand, it is moving in 5' to 3' direction. But since the strand is parental strand is 3' to 5', so here Okazaki fragments are formed. These particular fragments are called the Okazaki fragments. And here these are getting ligated. Here you can see the ligase enzyme. These are being ligated using the ligase enzyme. The DNA polymerase 1 is removing the primers, and DNA polymerase 3 is synthesizing the adding the new nucleotides. These are the primer which are getting formed by the with the help of enzyme primase. Okay, primase always forms the primers. Okay, okay. So topoisomerases are asked several times in the exam. So I already told you means explained a little bit about the topoisomerases. So what it does is, here I have explained you in a picture. So it trying to release the supercoil, so it will make a cut over here, it will again, and these two parts will be separated, isn't it? Again it will take this part and this part, it will join over outside, not over this molecule. So what will happen is, whatever torsional stress was created at this point, that will be released. यहाँ पे वो क्या कर रहा है कि ये उसके ऊपर ऐसे चढ़ा हुआ था ना, है ना, ये मॉलिक्यूल इसके ऊपर ऐसे पड़ा हुआ था, ये उसको जो भी वहां पे स्ट्रेस प्रोड्यूस हो रही थी, ये यहाँ पे कट करेगी, यहाँ पे कट करेगी, तो ये एक मॉलिक्यूल बन गया, ये एक मॉलिक्यूल बन गया, ठीक है। अभी ये दोनों को इसके ऊपर से नहीं लेके जाएगा, बस ये इस दोनों को जोड़ देगा बाहर लाके, इसके ऊपर उसको फिर से नहीं जाना, मतलब यहाँ पे जो भी स्ट्रेस क्रिएट हो रही थी वो रिलीज हो गई, ठीक है। तो ये काम करता है तुम्हारा टोपोआइसोमरेज. दिस इज द दिस इज द फंक्शन व्हाट योर टोपोआइसोमरेज विल डू. ओके, next, this is very important from the SRF point of view. The students preparing for SRF, so there are two classes of topoisomerases. Topoisomerase 1 works by forming the single strand cut, and topoisomerase 2 forms the double strand cuts. And both of them, topoisomerase 1 and 2, they both release the negative supercoiling. Okay, and the molecule that is topo 2, that is the DNA gyrase molecule is found in case of a prokaryote. Just now in the table we have seen that what it does is, it does not release the supercoiling, it generates negative supercoiling. Okay, and this process of generation of negative supercoiling is ATP requiring process, means active process. This is important that it needs ATP in the all the processes. Then reverse gyrase is the process. Reverse gyrase is a molecule that is found in case of your thermophiles. Means थर्मोफाइल्स में वो मॉलिक्यूल, वो ऑर्गेनिज्म होते हैं जो कि बहुत ही ज्यादा हाई टेम्परेचर पर रहते हैं, ठीक है। जैसे कि तुम्हारे हीट स्ट्रीम्स हो गए हैं, और जो भी लावा जहाँ पे निकलता है वो वाले जगह हो गए, वहां पे ये चीज रहते हैं, ठीक है। उसकी वजह से क्या होता है कि वहां पे बहुत ही कम चीजें सर्वाइव कर पाती है, बहुत कम ऑर्गेनिज्म सर्वाइव कर पाते हैं। तो नॉर्मल मॉलिक्यूल, नॉर्मल ऑर्गेनिज्म के पास जो भी मॉलिक्यूल होते हैं, वो मॉलिक्यूल इससे डिफर करते हैं, ठीक है। सो this thermophiles are found in the extreme climates, in the hot springs and lava producing places. So the normal molecule present in this particular organism will be differing from other similar kind of bacteria or prokaryote or eukaryote. Okay, so what it does is, it also generates the supercoiling, negative positive supercoiling. So all these three things, they have to deal with the negative supercoiling, but the reverse gyrase, this does the positive supercoiling. And what the DNA helicase was doing, DNA helical does the thing that it would unwind the DNA, and where it unwinds during the using the place where the leucine zipper is there. Leucine zipper question was asked 2 years back in SRF. I think in 2018. That time it was what is the leucine zipper was. I don't remember the exact question, but something was there on the thing, that is leucine zipper. So leucine zipper is a thing where continuously you can find the in the particular protein, you can find continuously there is a long stretch of leucines are present. Leucine is an amino acid, so long stretch of leucine is present. That particular thing is called leucine zipper. So in the comparatively means when you see the same structure in the DNA form, means from the stretch where the protein is getting synthesized. So the leucine encoding gene, leucine encoding the code on the okay. And next molecule is the DNA helicase. What it does is DNA helicase unwinds the double helix by breaking the hydrogen bonds between the complementary bases. Okay, here the two strands have been shown. Here it will just go and break this double stranded and break the hydrogen bonds between the things, and it will cause the unwinding of DNA. It helps in the unwinding of DNA. Okay. Okay. Then here I have again summarized the function of the topoisomerase helicase and single stranded binding protein. So topoisomerase, what it will do, you know already now. It will release the stress. And DNA helicase, what it will do, it will help the unwinding of the, it will break the hydrogen bonds between two strands, and it allows the two strand to form the replication fork. And single strand binding protein, it will help the strand to remain as such. Okay. Now coming to the other important molecule, that is the DNA polymerases. From this section also they used to in the name, and they used to ask the function. So this table is very very important. The tables I have shown in the today's class are, I have selected from various books, and only the chosen and very specific tables I have kept here. So all the tables are very very important. And first one is DNA polymerase 1 2 3 is shown over here. And in case of your prokaryotes, the 1 2 3 notation is given. In case of your eukaryotes, alpha beta gamma, like this kind of notation is given for DNA polymerases. And here the important enzyme is the DNA polymerase 3, which actually does the replication. And DNA polymerase 1 and 2, they are involved in some other activities. So DNA polymerase 2 is involved in DNA repair mechanism, and DNA polymerase 1 is involved in your gap filling and the removal of RNA primer. Okay. DNA polymerase 1 has both the 5' to 3' and 3' to 5' exonuclease activity. So it is used in the proofreading activity also. And it has this DNA polymerase 3 also only have the 3' to 5' exonuclease activity, no 3' to 5'. Okay. So from the particular DNA polymerase 1, sometimes we used to engineer several molecules in the using biotechnological tools. Okay. In case of eukaryotes, you can see over here this particular enzyme, that is the DNA polymerase sigma. This is very very important, and this is similar to that of your DNA polymerase 3, which actually does the function of replication of the DNA. And DNA polymerase beta and other things, they used to do the function of repair. Normally from eukaryotic DNA polymerase, they don't ask many questions. Still somehow have a look on this. And you need to understand the fact that DNA polymerase 1 2 3 present over the prokaryote. It will have the complementary thing in eukaryote also. Don't worry students, I think the topic might be a little bit hectic, but we'll be having a fun element in the very last. So stay tuned with me. Lecture is going to end very soon, in 15 minutes I think. Okay. So comparing the here I will not discuss all the points present written over here. I'll only explain the striking differences. So for recognition molecule that is present over in the prokaryotic cell, you can find the DNA A protein. But in case of your eukaryotic cell, there is no such, means something is involved, but that is completely unknown till now. And here helicase is unwinding protein, primase, all these things are same. The difference is over here, leading in the leading strand, the molecule leading and lagging strand, both the cases prokaryotes are DNA polymerase 3 is there. Okay. हाँ बांधी हुई. So in case of a leading and lagging strand, you can see that the DNA polymerase 3 is involved. But in this case, in eukaryotic cell, in leading strand, DNA polymerase sigma is there. In lagging strand, DNA polymerase alpha is there. Okay. And in the replacement of RNA with DNA in the region of the primer, here the DNA polymerase 1 is involved in prokaryote. Okay. DNA polymerase 1 is involved. And is unknown. Okay. And here in the prokaryote, you can find the molecule DNA gyrase, which is much more similar to DNA topoisomerase 2, which removes the supercoiling, removes the positive supercoils, and here the normal DNA topoisomerase 2 works. Okay. In the next section, that is the RNA molecules, RNA and the molecule associated with the various processes related to RNA. So this is the typical molecule of RNA nucleotide. Here you can find, I told you that in the second base you can find the oxygen OH group present over here, and this is the your phosphate. This particular thing is the phosphate, and this is the base. Okay. So that is the thing. Here you can see the RNA polynucleotide strand present over here, and it also runs in the 5' to 3' direction. And coming to the various types of the RNA present, and there are two types of RNA: coding and non-coding RNA. Coding RNAs are genuinely very less in number, and non-coding RNAs constitute the major section. And in the coding RNA section, mRNA is the only RNA which is coding, and the other RNAs are non-coding in nature. In the non-coding section, there are many RNA molecules are present, like rRNA, tRNA, snRNA, snORNA, all things there. Non-coding RNA molecules are actually important. Why? Because they assist in the formation, they assist in the regulation and various modulation of the processes involved in the mRNA. Okay. And mRNA and its expression. Okay. These two molecules, snRNA and snORNA, these two are involved in splicing process. Okay. And miRNA and siRNA, miRNA and siRNA, these two are involved in the process of your जीन रेगुलेशन. दे आर इन्वॉल्व इन द प्रोसेस ऑफ आरएनए इंटरफेरेंस. तो एमआईआरएनए और एसआईआरएनए क्या है? ये दोनों आपके आरएनए इंटरफेरेंस प्रोसेस में काम करते हैं. सो दे विल गो एंड फाइंड द प्लेस वेयर दे आर कॉम्प्लीमेंटेड टू ईच अदर, complementary sequence that is present in the RNA, and they will go and bind to that, and there is a specific enzyme which will cut that particular portion, or it will not let the RNA to form the protein. Okay. So in that way it is silencing the particular DNA, a particular gene. So the gene is not expressed. Okay. Among these non-coding RNAs, rRNA is found in the highest abundance amount. Among the three RNAs, common three RNA, mRNA, rRNA, tRNA also, RNA is found abundantly found, and rRNA, obviously you know that is the part of your structural part of your ribosome. Okay. mRNA is the coding region of the protein, and tRNA carries the amino acid or the particular chains while growing. And then small nuclear RNA also there, that is the pre-RNA, pre-mRNA, and from which the matured which is carrying the introns, and later on it will release the thing, that is the intron, and it will form the mature RNA. And sorry, my mistake, that is hnRNA. hnRNA is the nascent RNA which is getting formed. Small nuclear RNA actually helps in the processing of the pre-mRNA or hnRNA. Okay. And snORNA, or small nuclear RNA, involved in the processing and assembly of rRNA. And miRNA, siRNA, I already told you. Then the RNA polymer is present over the E. coli, that is, it is consisting of this particular thing, means the total enzyme, 2 alpha and beta dash, and this sigma molecule, whenever the sigma is attached to these four things, it is formed the functional thing, and when the sigma is getting detached, that is the core enzyme. Okay. Sigma molecules will help you to identify the point of promoter region where the RNA polymerase has to act upon. Okay. and eukaryotic RNA so you need to remember, you can remember it like RMT 1, 2, 3. So you will not confuse. So RNA polymerase 1 will form the rRNA, RNA polymerase 2 will form the mRNA, RNA polymerase 3 will basically form the tRNA. From these three things, it is important. Rest of the things you can remember. And here I have given the basic striking features between your transcription and RNA modification between your prokaryote and eukaryote. So you can see here the promoter region consisting of the consensus sequence, that is -35 and -10 sequence. That is the -35 region. You can -35 and -10 region. They are actually the region where your RNA polymerase will go and bind. It will recognize the particular thing. Okay. And in eukaryote, you can find the Tata box. Okay. And Tata box is otherwise called as Hogness box. In case of your prokaryote, you can find a single RNA polymerase doing all the activity, but in prokaryote, eukaryote, you can find three types of RNA polymerase doing all the different factors, different functions. Initiation is done by the sigma factor over bacteria, and here different factors are present. Here the termination is row dependent and row independent, but in case of your eukaryote, you can find it is allosteric model is there, because based upon which we can see the termination event. And here the capping and tailing does not occur. RNA capping and RNA tailing does not occur, but in eukaryote 7-methyl guanosine cap is added at the 5 prime, and the 3 prime end tailing poly A tail is getting added, that is tailing is occurring. Okay. Here I have shown you the various processes which is occurring. So you know that splicing का मतलब क्या होता है कि आपका जो भी mRNA नए नए बना हुआ है उसके अंदर आपका इंट्रॉन और एक्सॉन्स रखे होते हैं. ठीक है. इंट्रॉन्स जो है वो नॉन कोडिंग सेगमेंट्स होते हैं, उनका कोई काम नहीं होता प्रोटीन के अंदर, वो प्रोटीन के लिए कोड नहीं करते, तो उनको हमें हटाना होता है. तो स्प्लाइसिंग उसी प्रोसेस को बोलते हैं जहाँ पे कि हम इंट्रॉन्स को हटाते हैं. सो स्प्लाइसिंग इज द प्रोसेस वेयर वी रिमूव द unnecessary thing present over the mRNA, that is the introns. Introns don't actually code for any functional amino acids. Okay. And here I have shown the 5 prime capping and tailing. I already told you. So sometimes this is also important for the SRF people, where they used to ask like group 1, 2, and in which of the particular mRNA, which of the particular intron there is self splicing is seen. So, in case of your rRNA, that is particularly ribozymes, they used to go for self splicing, and mRNA lysosome formation, tRNA go for enzymatic splicing. Okay. Next is this, all of you know, I'm just skipping it. Let me tell you a little bit about this. So here we can find the differences in prokaryote and eukaryote on the basis of the various molecules present over the ribosomal structure. So basically in prokaryote you can find 50s, and in eukaryote you can find, in prokaryote you can find 50s and 30s. So total 70s molecule you can find, and in case of eukaryote you can find 40s and 60s. So 80s molecule you can find in case of your ribosome. Okay. And there are several exceptions present over the genetic code also. We found the several exceptions basically for particular amino acid in some, in case of your yeast, some case in case of your mammal, and like that. Okay. So the most important one among these is this three particular thing. Why? Because these are the stop codons normally, but they code for some particular things like glutamine in case of UA, UAG code for glutamine in case of protozoa, UGA codes for selenocysteine in case of your some bacteria, and UAG also codes for some of the pyrolysin molecule in case of your methane producing bacteria. So these are the peculiar things. So you need to somehow remember this. Okay. So since time is less, I'll be just quickly briefing this. So bacterial factors, if you just see the translation transcription factors, sorry, the heading is a little bit wrong. This is the process which facilitate, means which actually responsible for your translation. So IF1, IF2, wherever things are there, all those things are basically for your bacteria. And EIF, wherever prefix is present over there, that is the eukaryotic factors. Okay. And here the important thing, if you compare the prokaryotic and eukaryotic translation, you can see here the initial binding requires the signed diagonal sequence, and here the initial binding requires the 5' methyl guanosine cap. Okay. And elongation rate is comparatively high here in the bacterial cell, that it is comparatively slow. And one of the striking feature present over here is that in case of your prokaryotes, you can find simultaneously production of your RNA and simultaneously going for translation. Means transcription translation goes hand by hand. Immediately they go for translation. But in case of your eukaryote, you cannot see after the formation of RNA, it has to go for some certain processing. Then only it can go for further translation process. Okay. Here I have listed some of the bacterial translation inhibitors like erythromycin, puromycin, tetracycline. In some of the exams they used to ask like what is the mechanism behind that. So just for example, tetracycline blocks the blocks elongation process of the translation. And likewise some of the important molecules under translation. Then there are several kinds of post translational modifications like histone acetylation, methylation. I told you. So here these two rows are very important, like acetylation and methylation. So here whatever acetylation, methylation is happening, that is occurring in the lysine residue. Okay, that is important. Next molecule present over molecular genetics is your restriction enzyme, which cuts the DNA at a segment, recognizing the particular segment, and it has three classes among them. The most important class is class 2-1, which actually recognizes the palindromic sequence and cuts it, and that is requires the Mg plus 2 for its functioning. But other two things they don't cut like that, and there is the significant difference between them, that these class 1 and class 3 are ATP dependent, but this one is Mg plus 2 dependent. Okay. Coming to the last part of today's session discussion, that is apart from all these molecules, there are molecules like methylases which actually add the methyl group, kinases which actually add the ATP, phosphatases which remove the phosphate group, and and then cyclins are important for your cell cycle progression. And exonucleases what it does is, sorry, kinases actually adds the phosphate group. So that is the thing, breaks the ATP and adds the phosphate group. Exonuclease actually cuts the DNA from the outside, and if we keep on listing, there will be so many number of molecules and associated with it. So we will limit today's discussion with this much molecules only. Okay, so there is a small quiz. It will take only 2-3 minutes. Hope everyone will join it, and it will be conducted online. Okay. So for attending the particular quiz, what you need to do is, sir, please share the link. Hello, sir.
Yes, I will share that link now. I think, what is that? Some reviewing.
Just a second, sir. Just a second. Let me open it.
Yeah.
Don't worry, students. I'll be answering your questions in a bit. I know you all have become tired. So to freshen you up, I have organized this quiz. Okay. So to attend the particular quiz, what you need to do is, you are given with a link in the chat box, isn't it? Click on the particular link. Why am I so confused? Okay. Have everyone got the link?
Wait. I'm putting now. Yeah, I have posted. Please check, students. I have posted one link.
Yeah, students, just click on that link. That link will don't means just open another tab and go for clicking this link. Don't move away from Zoom. Just click on that link so that you will open a tab in the Chrome or whatever browser you are using.
Well, students, some students were asking we need only materials, but students, remember the material preparation is based on completely on classes only. So getting study material itself is not worthy. Classes and writing exams are a good worthy for you. And I have put the PDF of the details. Please make note of it.
Did you join, students? So first thing is, what are the terms and concepts you learned from today's session? Anything? Okay, molecular genetics, basic concepts. Then answer faster, please. Histones. Okay. Okay, we'll move to the next question. DNA, RNA. Okay. Join faster. Join faster. There are 33 students online, but only few have joined. Join faster. Yes, quickly. Trust me, it will be fun. Join faster. Okay. Okay, I'm starting the quiz. Central dogma concept was given by which scientist? You need to answer Watson, Crick, or Beadle and Tatum. Who given the concept of central dogma? Click on the answer. Time's up. So 7 of the students have answered that it is Crick. Yes, it is, the answer is Crick. Central dogma concept was given by Crick. Moving to the next question. Okay, so leaderboard is present over here. The first students, in what speed you are answering the question, that also depends, and that also decides whether you will be in what position. So Subrajyoti Nayak gets the first one, first rank, then Meghna, then Naveen, then Harsh, then Akanksha, Siddhant. Okay, move to the next question. Histones are alkaline in nature. Is it true or false? You need to tick that. Answer faster. Okay. Yes, it is true. Histones are alkaline in nature. Let's have a look at the leaderboard. Okay, now hers has raised up in the table. Now we have the new winners. Let's move to the next question. Acetylation of histone inhibits the expression of the particular region. Is it true? Acetylation of histone inhibits the expression of particular region, or methylation does so? What is the answer? Okay, time's up. Half of the students have given true, half of the students have given false. So acetylation, I already told, acetylation will loosen up the DNA. His acetylation क्या करेगा कि आपके जो DNA हेलिक्स है, आपका जो हिस्टोन मॉलिक्यूल का न्यूक्लियोसोम का जो स्ट्रक्चर है उसका जो कॉम्पैक्टली बाइंड होकर रखा होगा उसको वो लूज करेगा, so that what will happen is transcription factor and all the molecules and get access to the DNA. वो जाके उसके पास पहुंच पाएंगी तो आपका इनिबिशन नहीं होगा, वो एक्सप्रेशन को मदद करेगा. मतलब अगर एसिटाइलेशन कहीं पे हो रही है तो वो दैट विल प्रमोट द एक्सप्रेशन. ओके, सो दिस स्टेटमेंट इज फॉल्स. दैट इज द करेक्ट आंसर. ट्रू इज द रॉन्ग आंसर. मिथाइलेशन जो है वो आपका एक्सप्रेशन को इनहिबिट करता है. सो द लीडर बोर्ड इज चेंजिंग. Okay, we have new winners, Meghna and Harsh. Then Subrajyoti. Subrajyoti has come down. I think he has not answered this question. Okay. And Aarti was fastest for this particular question. RNA polymerase is responsible for synthesis of tRNA. Which RNA polymerase synthesizes tRNA? Right now only, few minutes back I told you this particular thing. Which RNA polymerase is responsible for tRNA? RNA polymerase 3, that is the correct answer. Okay. Okay, Meghna is the overall winner. Congratulations, Meghna. And then Harsh and Subrajyoti. They have also played well. Everyone played well. This was the first time means we were using this particular platform. I think it was fun. Okay. So we'll keep on using this platform if your people like it. Okay. So there is the last question. What do you want to say about today's class in your words? Just tell me about a review. I'm asking regarding, do you like the class? Which section you didn't like? What what other thing you want to be there in the class? Good, thank you. Okay, I like it. Okay, impressive, effective. Details. Okay, you want the details. Definitely, details will be there in the regular classes because there was time limitation. Details was not there today. I wanted to comprehend the several things so that that will be a revision for you guys. Awesome, impressive. Liked it. Thank you. Thank you so much for all your review. I will try to implement whatever things you have told in the subsequent classes. Clear explanation you want? Definitely, because today there is a little bit of internet fluctuation was going on. I was also a bit disturbed today. Okay, thank you. English fluency is good. Class was bit fast because we had this much things to cover in limited time. No, that's why it was a bit fast. Okay, it was not fantastic to you. Why you didn't like it? Is there any particular reason? You were still confused. Okay, I think you need to learn the concept, and because I have merged so many things, I think that's why you might have become confused. Definitely, we'll include the SRF points and JRF points definitely, and network issue is there. We'll be definitely trying to solve the thing. Okay. Basic terms will be reviewing definitely in the detailed classes. Will be reviewing the basic terms, whatever. Okay. Thank you so much for all your responses. Right now, I'll be seeing whatever chat boxes, chats you have made, and whatever queries you had, I'll be answering them. Since you all might have tired, just give me 5 minutes. I'll answer all the things.
Well, students, someone asking about we need a clear explanation. Yeah, we will give clear explanation because this is a demo class. We have to cover many things from A to Z. We have to cover many things. That's why we are running some bit faster. It's only for basic understanding to show how we explain to you how we provide a quality of education to you. That's all. In regular classes, you will get everything, and the classes will be very clear, crisper, and you will get an individual discussion will be there. Thank you.
Okay, the very first question was by Aditya. He asked about why ZDNA is formed there. I think he's talking about the section where I told you that unwinding has occurred, and at that position ZDNA is formed. Why ZDNA is formed there? Because see, in the normal DNA, BDNA, it is right-handed coiling, isn't it? So because of unwinding at the particular region, definitely if it is right-handed, if you try to unwind something, the opposite direction you have to move, no? So because of that reason, what the force has, what external force has did is it will unwind, and because of that unwinding it will naturally form a left-handed coiling. So because of that transiently we can see the ZDNA. आपको समझ आ रही है? क्योंकि BDNA जो होता है वो नॉर्मली क्या होता है, वो राइट हैंडेड स्ट्रक्चर होता है. किसी भी राइट हैंडेड जो रोप होता है कोई भी, उसको हम अगर लूज करना चाहते हैं तो अपोजिट डायरेक्शन में हमें घुमाना पड़ेगा, राइट? ऑपोजिट डायरेक्शन में घुमाएंगे तो ऑटोमेटिकली राइट हैंडेड कॉइलिंग जो था उसको ऑपोजिट घुमाएंगे तो लेफ्ट हैंडेड कॉइलिंग तो आएगी. तो लेफ्ट हैंडेड कॉइलिंग किस में आती है? वो तो डीएनए का डीएनए ही पड़ा हुआ है. तो वो जब लेफ्ट हैंडेड कॉइलिंग हो गया, दैट विल फॉर्म द ZDNA, बट दैट थिंग इज ट्रांजिएंट बिकॉज दैट इज फॉर्म बिकॉज ऑफ ओनली द ओपनिंग ऑफ द fork. So that ZDNA formed in that particular position is not permanent. Okay. Then the next question is, I guess there are no more questions. Okay, what is housekeeping gene? Somebody has asked. Housekeeping gene is the gene which is synthesized continuously. Okay, it will not means some genes are there which are synthesized and which is expressed in the particular kind of situation only. Like say the molecules which is required to heat response that are expressed when the heat is provided or heat shock is provided. At that time only they will express. Okay, that gene are not housekeeping gene. Housekeeping genes are those which are continuously synthesized and continuously whose product are continuously used by the cell. For example, your histones are regularly required by the cell to package the DNA, newly formed DNA. And your rRNA molecules, rRNA producing genes are one of the housekeeping genes because they need to be constantly formed, and those genes are required in high amount in the DNA. Because of that particular region, they are called as housekeeping genes. Definitely we'll be organizing such quizzes in the end. Definitely. Is there any more doubts? Okay, I guess no doubt is there. I think today was a very exhaustive and why I took so many topics together is because so many of you are having the upcoming exam. And to the beginners also, if the term is going to your ears, means एक बार अगर आप सुनते हो terms को, तो अगर एक बार आपके दिमाग में चला जाता है कि हाँ हमने ये term सुना हुआ है, we have heard of this term sometimes. So whenever you will read the book or whenever you will get across the particular term or explanation, you will get to know better. Okay, it might be true that you might have not understood everything whatever I taught today, but you would have got something brief idea about the topic. So whenever you are going through the book, you will get to understand the better. Okay, and thank you for your patient hearing, and whatever suggestions you told, I'll definitely implementing them in the upcoming classes. Thank you.
Thank you, students. I hope that's all for today's class. Thank you, students. You may leave the channel. Thank you.